is missing. You can see it. So that's this individual produces. This is just the test cross you're bringing in.
You're like, oh, these little bundles of jelly.
Because it's in. So they're not going to sort of jump on that. Just imagine. Almond layer. You want to separate them. How are you going to do that? You'd have to kind of cut this off. Since they put together on the same physical entity, one side. That's the simplicity. Oh, the Himalayan frog? I can find it for you.
Touch every. Is that it kind of rehydrates you?
the bus, they were just like, some girls like, your food looks really good. And I was like, oh, thanks. But I was like, why do you complain? I'm like, oh, bye. Well, this is yours, right? Yeah. I was like, that's my pen, but I was like, this is my notebook. I literally just ran into I swear. Hey. Yeah, I was just like, hey. And then we opened the door and it was like, here. And I was just like, hey. I was gonna study this weekend, but I think I won't because I have Spanish to study. And I'm actually like, I'll help you with Spanish.
The kids, it was basically this girl who traveled to all those like.
I need to like read all of the like short films
That was like what she did, and then she
and
created.
writings and whatever we talked over in class. So that she's gonna be on the midterm, which is so stupid. Why are you testing us on our memorization? Like what happened? What the fuck? You should test us on like grammar and like writing skills. Yes, I do actually. I want to try. Yeah. Was it this? Yeah. It looks humongous. I thought it was like, it is. It's a big ass platter. I think strawberries are included. It's really good.
It's actually a nice.
She has a lot. And I was telling her, I was like, I see it on the bus, and everyone was just staring at it because it was like this big, and I was just my baby. She's gonna cut her. You have to get back. I told him, hey, we should do this. We should like train for the Boston Marathon. Like, he's like, I don't know. I'm like, so fucking dumb. You are so stupid for thinking that. And like, usually I'm the one that's scared. He's like, I'm just gonna. And he's like, why are you scared? He's a chicken. Like, he's such a chicken. He's like, you can. And like, he always just like, you're such a fucking because he called me a kid. No, it's not scary, like, because I was thinking about that sort of example something, but then, like, I just, like, like, recently one of the, like, don't say that to me. One of the people who had done it a billion times, like, the trainer is like,
Okay, so now let's actually look at the actual experiments that were done and the actual results that were obtained by Morgan. So Morgan performed the following cross in Drosophila to investigate independent assortment of genes. And he was looking at two different genes, purple colored eyes and vestigial wings. So the normal eyes are red. The mutant eyes give you purple colored eyes. Normally you have nice big wings in the fly, and then you have a mutant that gives you tiny little stubs of wings, vestigial wings. Okay? So he started a cross where he had a fly that was true breeding, normal colored eyes, normal sized wings, and he crossed it to a fly that had purple colored eyes and vestigial wings, true breeding. And then in the F1, he got normal colored eyes, normal colored wings, or normal sized wings, heterozygous, and he then decided to do a test cross. So he took the PR plus VG plus PR VG heterozygous F1 fly, crossed it to a homozygous recessive fly, PR VG, PR VG. And then he investigated what happens. Now, if these two genes were on two separate chromosomes, according to Mendel's rules, they should assort independently of one another, correct? So in theory, if they are on different chromosomes, they should assort independently in this test cross, and you should get 4 different gamete classes, PR plus VG plus, PR VG, PR VG plus, and PR plus VG. And you get 4 different phenotypes in the flies. PR plus VG plus, which would be red eyes and normal wings. PR VG, which would be purple eyes and vestigial wings. PR VG plus, which would be basically purple eyes and normal wings. And these would be normal red eyes and vestigial wings. 4 different classes. If these two genes are on different chromosomes and are assorting them independently, the ratio of these 4 phenotype classes would be 25%. Correct? If they are on the same chromosome, as I showed you just previously, and they are right next to one another on the same chromosome, what would you expect to get? You would expect to get, remember the cross, the parent was PR plus VG plus and PR VG, correct? So based on the previous slide, what you would expect to get in the F2s would be 50%, 50%, and these classes would be missing. Correct? Yes? Because this PR plus and VG plus are linked on one chromosome, PR VG is linked on the other chromosome. Yes? Understand? Somebody's shaking their head. No?
How would you respond to the question? Would you think the ratio on
Sorry, how would I what?
how would you respond to the question? Would you give us a ratio and figure out why we can the ratio of 17?
I'll give you these ratios. You wouldn't have to figure out these ratios. There's no way you can figure that out. Except by doing the experiment, sorry. There are ways to figure it out. Just you have to do the experiment. Yeah. So that's what I would ask is, are the genes linked? That's the question. And to figure out if the genes are linked, we look for this difference. You get 4 classes, equal numbers or unequal numbers. Right? That's what you would say. All right. So what did Morgan actually find in this experiment? When he did that experiment, here's what Morgan found. 1,005 flies were looking like this spell, 968 looked like this spell. But he also got 143 that were like this, and 153 that looked like this. So it's not 0. It's not 50, 50, 0, 0. Correct? The ratio is actually 44, 44, 6, and 6 on average. Correct? So what's going on? Why is it that you're not getting the 0? You're close to 0, right? You could round it up and say, okay, it's almost 0. But it's not 0. And what's happening here? Question. Sorry? Crossing over. Exactly. So what's crossing over? That's where we're going next. So although the non-parental class, this is known as the non-parental class, these are known as the parental classes because this class looks like this parent, this class looks like this parent. So this class here is called the parental class. These classes here are called the non-parental classes or the recombinant classes. Okay? Two interchangeable terms, recombinant, non-parental. So the non-parental classes are present, but the frequencies are dramatically reduced. Okay? And the two loci are linked, but they're not completely linked. So the theoretical explanation would have been that they're completely linked. In practice, they're not completely linked. They're linked, but occasionally they break apart and separate. And how do you explain the non-parental classes? And the answer was given to us. It's crossing over. So what happens in meiosis 1? All of this happens during meiosis 1. So here is the chromosome. Here is the PR gene on the chromosome. Here is the VG gene on the chromosome. There's your parental chromosome. And you cross it to a homozygous recessive PR, VG, PR, VG. What happens in meiosis 1 is these chromosomes pair. Okay. So in the heterozygous condition, these chromosomes will pair. The PR plus VG chromosome will pair with the PR VG homolog. And then, occasionally, you get a crossing over. You basically exchange physical genetic material. And you end up with chromosomes at the end of meiosis 1 that are one sister chromatid is PRVG. The other sister chromatid, because it's exchanged one of these arms, becomes PRVG plus. And it's a reciprocal exchange. So the other homolog, you have PR plus and VG, and then PR plus VG plus on the other sister. Yes?
On the last slide, was there a typo on the third row? The third row, isn't that P supposed to be lowercase?
Third row.
In your diagram.
Here? Oh,
From the top bottom third row, the PRVG,
PR, this here?
to the left of it, is it supposed to be lowercase?
Yes. Yes. This is auto-formatting itself. Okay, so these are now crossed over chromosomes, or chromatids basically. They've exchanged genetic material, and it's because of that that you will get the recombinant classes or the non-parental classes. So the chromosomes have gone through crossover, and the crossover products are called recombinants basically. And the original chromosomes that have not undergone crossover will be the parental chromosomes. And we've seen this, obviously. We looked at the experiments of Barbara McClintock with the corn, where she had a chromosome with a knob and a tail, and she was able to show how crossing over took place with physical exchange of genetic material. So if you go through your notes on meiosis, you will see that there was an experiment I discussed from Barbara McClintock in the 1930s using corn, where she showed that crossing over was actually an exchange of physical genomic material. During meiosis 1, when the homologs pair together, the non-sister chromatids cross each other. And the resulting cross-shaped structure called the chiasmata results in an exchange of chromosome arms. So if you look at crossing over under the microscope, here is one chromosome, here is its homolog. They've undergone replication already. Right? These are duplicated homologous chromosomes. They pair like this. They synapse in meiosis 1, and then they exchange. Little fragments get exchanged right here and right here. There's a crossover taking place at this point. So crossing over takes place between homologous chromosomes, but between non-sister chromatids. This chromatid is not the sister, it doesn't exchange with the sister chromatid, it exchanges with the non-sister chromatid, with the chromatid on the other homolog. And so then, crossing over between non-sister chromatids, and you end up with a chromosome, in end of meiosis 1, you end up with a chromosome that has this little fragment and this little fragment exchanged. Then they go into meiosis 2. This is anaphase meiosis 1, segregation of homologous chromosomes. And then you go into meiosis 2, you get one chromosome that looks like the parental arm, completely black. Another chromosome that's almost completely black, a little bit of the gray at the end, but crossing over took place. Third chromosome that's almost completely gray, with a segment that's black because it's exchanged it with this chromosome here. And then the fourth chromosome that's completely gray. So this chromosome looks like this chromosome, the parental chromosome. This gamete looks like this gamete from the parent. And this gamete is a recombinant between the two parents, and this one is a recombinant between the two parents. And so that's why they're called recombinant gametes or recombinant individuals. Yeah? So, haploid gametes in meiosis 2. Now, of course, these gametes will get fertilized, and you will get a progeny, and then you'll analyze the progeny by looking at the phenotypes. So how does one determine whether two genes reside on different chromosomes or reside on the same chromosome? And they are linked. So that's the question. That was the question you were asking as well earlier. So we need to define a term. We need to define parental and recombinant. And you guys need to clearly understand what is the parental and what is the recombinant phenotypes and gametes. Okay? So it depends on the true breeding parent when you start a cross. If I start a cross with a parent that's big A, big B, true breeding, cross to a parent that's little a, little b, the parental phenotype or the parental gametes, one parental gamete will be big A and big B, and one parental gamete will be little a and little b. Okay. Here is your parental big A, big B, parental little a. They will get fertilized to produce a heterozygous individual, big A, big B, and little a, little b. Now you have a heterozygous condition. They will produce gametes, meiosis, following this 4 different gametes, big A, big B, big A, little b, little a, big B, and little a, little b. And these, little a, little b, and big A, big B, these are your parental gametes. These ones here, big A, little b, and little a, big B, these are your non-parental gametes. They don't look like your parents, and they are, in effect, recombinant gametes. So the recombinant gametes are those with different allelic combinations than the gametes of the previous generation. So you look at the gametes in the first generation, big A, big B, and little a, little b. Big A, big B is a parental gamete. Big A, big B in the second generation is the parental gamete. Little a, little b is the parental gamete in the first generation. Little a, little b is the parental gamete in the second generation. The gametes that are not present in the first generation are recombinant gametes, basically. And the progeny that they produce will be recombinant progeny. Okay? So, genes that are located on the same homologous chromosomes called linked genes. And so when big A and big C alleles are introduced from one parent and they are physically linked on the same chromosome, they are called linked genes. So, you take a heterozygous condition where you have big A, little a, big C, little c. It depends on how you have them together. This is known as coupling. Big A and big C in one chromosome when they're linked, or little a and little c on one chromosome if they're linked, is known as the coupling condition. Coupling condition means two dominant alleles are on the same chromosome or two recessive alleles are on the same chromosome. That's known as the coupling conformation. So, now if you have a condition where you have a parent where you have big A linked to little c on the same chromosome, or little a linked to big C, this is known as the repulsion conformation. So we're talking about the parental conformation in the original cross, the original parent. In the original parentage, if you are big A with big C or little a with little c, those two genes are coupled together. If you have a big A gene coupled to a little a gene, a little c gene, and a little a gene coupled to a big C gene, then those two genes are in what's called the repulsion conformation. It's just a terminology. I try not to use it in the midterm if I can avoid it. It's not something you need to completely memorize. If I have used it and you've forgotten it, come and ask me. Okay? I will basically tell you. So when you have linkage of dominant and recessive together, it's called repulsion. When you have linkage of two dominants or two recessives together, it's known as coupling. Yes? No. These are homologous chromosomes. True breeding parents. Correct? So I'm just showing it to you. This is not in a single individual. I'm just showing. If you want to talk about sister chromatids, let's start with an individual like this. Okay? Here is one chromosome. Here is the homolog. And if you are big A, big C, and little a, and big A, big C, this is coupling. This is the parental generation. Okay? Big A, big C, big A, big C, true breeding individual. Here is another true breeding individual. Homologous chromosomes, little a, little c, little a, little c. This is also coupling. Right? If you now look at an individual, true breeding individual, that's big A, little c, big A, little c, parental generation. This is the repulsion conformation. Or you can have a parent that's little a, big C, little a, big C. That's also repulsion conformation. Correct? So, if A and B are on different chromosomes, you're going to do a cross like this. That's the parent. This parent will generate gametes that will be big A, big B, and little a, little b. These are your gametes, correct? This parent here will produce big A and big B gamete. This parent here will produce little a and little b gamete. And you get an F1 diploid. And that F1 diploid will be big A, little a, and big B, little b. Now you're going to do a test cross, little a, little b, little a, little b.